blood on your plate like that, what do you do?
Hold it to the light.
Hold it up to the light so you can see the correct hemolysis. There will be an area circled and an arrow to it. Not everything on the plate, not somewhere else, but whatever's circled. Okay? That would be the section you might have to identify. And this blood on your plate is going to grow pretty much almost everything, because blood is actually an enrichment medium. It's not selective. I guess you could call it complex/enrichment, but it is or is not differential. Differential media means to be able to tell the difference on the plate.
Is that differential?
It is differential, not selective. It's differential because you can tell groups of organisms apart by alpha, beta, and gamma hemolysis. Gamma streptococci. Do you worry about it? It means no hemolysis, or just stereococci. They're not going to hurt you. Gamma means no hemolysis at all. They're just the foods, and they help you. Can y'all hear? Way back over there. There's a section up here. That plate. Know the media. I'm not going to have it labeled, of course. What plate is this? Tryptic soy lager. Complex media or selective? Complex means grows everything. Grows almost everything. There's no such thing as grows everything. Complex. Everything grows. Or almost everything. You streaked 2 laboratory cultures on this. What were the 2 organisms that you streaked from 2s? Staphylococcus epidermidis, Streptococcus, also called Enterococcus faecalis. Now it says in your lab manual Streptococcus enterococcus facial. If you say one or the other, you're okay, because they are like the closest cousins you could ever get. They're going to give you the same result. Both grew. Staphylococcus epidermidis, Streptococcus, which is now called Enterococcus faecalis. In the lab manual. Now, streptococci often are fastidious organisms. I said often, I say every single one of them. That means they're what? Picky eaters. So if you have a picky eater, do they tend to grow as well as something that will eat everything? No. One of these types of organisms is opaque in the colony and one is translucent. Who's opaque? Staphylococci. The streptococci/enterococci are often picky eaters, so they give you translucent, which is less heavy growth. Also took this plate and you placed on it a reagent. What reagent did you put on both sides of this plate? Hydrogen peroxide. What test was that?
Catalase.
Not oxidase. What test was that? Catalase. Catalase is an enzyme that breaks down hydrogen peroxide to water and oxygen. Oxygen is a bond. Which one was catalase positive? Which one was catalase negative? Staph was positive. Strep was negative. So you already have 2 ways to tell staphylococci from a streptococci. You do have to spell out staphylococci and streptococci, because I know what they are. So I've got to be sure you know what they are. You have to spell it out. Don't just put an S. Don't even just put staph or strep. Spell it out. Okay? Next plate. Pink. Artificial pink. What type of plate is this? Mannitol salt agar. Mannitol salt agar. Now, one of your cultures grew and one did not. But you streaked from the master culture. What grew? Staphylococcus epidermidis. Because salt tolerant. What? Because you're always in that whole lab was telling a staphylococcus from a streptococcus to differentiate between a staphylococcus and a streptococcus, now called enterococcus. What did not grow? Streptococcus did not grow. Streptococcus faecalis, enterococcus faecalis. Well, also had, this did not come from a culture I gave you, but from a body. This has to be, again, a staph because this media is complex or selective. Selective. The salts make it select for staphylococci. Now this is a staphylococci too. This is a staphylococci. You see the difference? This one is epidermidis. Who is this? Probable. Probable Staphylococcus aureus. You don't have to say probable, but in reality it's presumptive Staphylococcus aureus. Staphylococcus aureus can be a wound infector. It can also cause toxic shock syndrome and other things. We did a test for this. Took a little bit of the culture, put it into rabbit plasma, and this is another presumptive test for Staphylococcus aureus. Staphylococcus aureus, if it's present, can produce what enzyme? Coagulase. What does the enzyme coagulase do? Doesn't break down. What does the enzyme coagulase do? It coagulates. It causes a fluid to move to a gelatin. Would this be negative or positive?
Negative.
Negative. Would this be negative or positive? Positive. If you saw this, what organism is indicated? Staphylococcus aureus. Okay? And of course the test is coagulase. Please, 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 please don't mix up catalase with coagulase. People do that every single semester. I think it's because they sound alike and they both start with a C. But don't mix them up, okay? Alright? Try to work on that one. Practice on that one. Okay. Mannitol sauce agar. Keep moving when we look at it. Mannitol sauce agar will select for staph, stop, strep, / enterococcus. The flip side, our plate that does the exact opposite, is blood agar crystallite. Blood agar crystallite. So blood agar crystallite would let what grow would stop what would grow. The strep and the enterococcus to grow is the crystal violet. And the crystal violet would stop the staph. Learn your mannitol salts plate because salts relates to salt-tolerant staph that can survive. Think of this one as the exact opposite. Okay? Now, if your blood agar crystal violet came out of that candle jar and you had, and also have it for your gnome, beautiful royal blue colonies, that's some sort of streptococcus species because it picks up the what dye? It picks up the crystal violet. The crystal violet's what? Violet, blue, purple. All the other colonies, if they grew, still it wouldn't be staph, but it could be something else. Okay? That's only from your throat. From the gnome cultures, you would say it stops the growth of staph, allows the growth of strep.
Does any strep that turns royal blue?
Any strep. Any strep will turn royal blue. Who's this? Chocolate agar. You won't have a CA on it, okay? Chocolate agar is what? It's blood agar that's been heated up. We're heating it up so the enzymes will not break down growth factors needed for, here we go again, fastidious organisms. Some of these could be strep, but we were doing the what test on this? The oxidase test. Do you remember this? That was the tube I broke to put the enzymes down to do the oxidase test right up to the chocolate agar. If the organisms, the colonies turned black, it was, well, presence of non-pathogenic who? Neisseria. Non-pathogenic Neisseria. With the oxidase test. Oxidase test turns the colonies of non-pathogenic Neisseria black because it detects certain cytokines. Alright? So you already know two reagents right now. Oxidase, hydrogen peroxide. This is for catalase, of course this is for oxidase. And the coagulase test is just rabbit plasma. What's the order of the brimstone? Crystal violet, grams iodine, ethanol, or ethyl alcohol, saccharine. The answer grams iodine, ethanol, saffron. Okay. Gram positives are what color? Purple. Gram negatives are what color?
Pink.
Pink. It's very important because it drops almost all bacteria into 1 and 2 groups to help identify eventually so you can give the right medicine to the patient. Okay? Now, you did what selects for staph, what selects for strep. I want to switch. I'm also trying to bring back things that were a long time ago because they're still there.
No, the oxidase says it's just squeeze that little tube that she snaps. What is it? She just called it oxidase. I don't know.
MRSA? Double check.
Is that what she just
Okay. MRSA you need to know though because it's a subgroup of Staphylococcus aureus. Okay, so the yellow plate would be Staphylococcus aureus. MSA. But we wouldn't know if it's MRSA unless I put it onto a plate with the antibiotic methicillin.
No, that was cool.
But you still need to be able to spell out MRSA, which is a subgroup of Staphylococcus aureus. So think about it. MRSA. SA is Staphylococcus aureus. R is resistant Staphylococcus aureus. M goes both to the original antibiotic resistance and what it is now. It would be methicillin resistant Staphylococcus aureus, first antibiotic, or what it's become now, multiple resistant Staphylococcus aureus.
We put multiple in the bank.
You put multiples okay, but you got to be able to spell everything out, all the way out. Are all MRSA Staphylococcus aureus? Yes. It's like all collies or dogs. Are all Staphylococcus aureus MRSA? No. No. Are all dogs collies? No. Okay. Sort of helps sort out the way you can see that. Okay, ask me questions. I'm not trying to race through this. I'm trying to get as much information as solidly to you as I can. Remember the parts of the scope. First, what type of scope did you have? Compound light microscope. Don't drive me nuts and put down electron microscope. Every year, I think somebody sacrifices 2 points or whatever for the partial credit because they just like to do that. There's no electron microscope. Pull it out of a cubby hole and put down at your station and plug in to look at. Okay, compound light scope. Know the parts. I have 2 practice questions again. One here is on parts of the scope. I understand you probably know the parts of the scope. The concern people have is they have to hunt for the little labels. And sometimes it's like, I can't find a label, I can't find a label. So practice finding the little labels so you can label the parts of the scope right here. The other practice question is back here. And it's mannitol salts. And those would be the parts of the scope. First you have the white, the inner ring, inner knob is your fine focus. Outside of it is your coarse adjustment. Nobody does anything but maybe some fine focus. If not, call me over. If you move that slide, it's going to probably, I may not be able to find it in time for the midterm. Okay? I spend 4 hours setting this up. I hunt for the best place on the slide for you. Don't move it. Don't move it because you're hurting everybody. Okay, next part, you have your stage. Well, that's where everything is on. Inside the stage, if you look, and this makes sense because like submarine within the water, within the stage is a piece of glass. That is to help focus light, well, obviously, light coming up into the objective lens. It's called the substage, like submarine. Substage condenser, because what does it do to the light? Focuses it up to the objective lens. You have an iris diaphragm that can open to give you more light, closes to give you less light. That's that small little knob on the front. Remember that. You have your objective lenses. Yes, you don't just say objective lens. You tell me the 4x, you tell me the 40x, you tell me, well, 4x, 10x, 40x, but actually some people have 45x, and the 100x. Which one's the old version? 100x. Which one do you use for bacteria? 100x in most cases. That's not the only lens you have. This is some magnification on the By the time you look through ocular lens, what's your total magnification once the light goes through 10 lenses? 1,000. Do you understand that? You go 100 times 10 gives you 1,000 total magnification. If you look at the 4x, by the time it gets to your eye, you're looking at 4 times 10, 40 magnification. Okay? So remember this, and please don't tell me that's an electron microscope. There are parts over here to try to find where the labels are. The labels, by the nature of what I'm trying to label, can't be big. Alright? So pay attention to that. Coming back, let's compare. I do not want you to close. Coming back over here. This is an electron scope. We already said no. Know the parts. Estimate the length. We've been over that before. I'm going to go over the stuff we haven't been over before. I'm going to go over things, topics on the board, and then you are going to have 30 minutes to look at slides, look at the test questions, and you personally to ask me questions about what you would like to know. Okay? 101. Where's the step 20? And you need to be able to spell spectrophotometer 20. It measures the constant. Huh? I'm sorry. Turbidity. I didn't get to finish the question. And how does it, that's good. How does it, what's the basis for measuring the amount of bacteria? Turbidity. Good going. Okay.
Can you spell it?
Okay. What it means is spectro is spectrum like light. Photo, light actually has packets called photons. It can be a wave or a particle when we look at physics. Meter means to measure. I don't know why the 20 is there. I have no idea why they put the 20 in. And it's the 20th machine that they did it. I don't know. Okay? All right. Remember, to be able with the graphs, you don't have to plot the graphs. But you do have to be able to recognize and label X and Y axis according to what I'm teaching. You actually can change the axis on the graphs a little bit and go by what I'm teaching. Be able to know the standard curves we went over last time for optical density and percent transmission that were read off the spectrophotometer 20. Okay, honestly, spec 20, but don't write down spec 20. Ground stains we already did. Stains. Positive stains and negative stains. We'll do this way back. The easiest way to do this, but yes, you need to know all of them, is learn the negative stains that we've done in lab and say everything else is positive stains. Two negative stains are Congo red, nigrosin black. Congo red and nigrosin black. What do they stain? The organism or the background? They stain the background. Positive stains stain the what? The organism. That's crystal violet, saffron, caramel fuchsia, non-pipe green, ethylene blue. Pretty long list. Now moving to the spore stains that you did. You had to drive for the Schaefer-Fulton the stain in with something into the spore coat. What did you use? Heat. What was the stain you used to stain the spore in the Schaefer-Fulton? Non-packed green. So what color is the spore? Green. You then use a positive counter stain to stain the bacteria. What was the counter stain in the Schaefer-Fulton? Saffron. Yes, it was red. So the cells are pink or red in a Schaefer-Fulton. If I have a pointer or something and ask you what are the green, what are the structures that are the green ovals, you would answer they are spores. If I have a pointer on a vegetative cell that's pink and ask what is this, you say bacilli. Yes.
Like, this says saffron. Is that
That's right. Yeah, that's saffron. I come from different places. Everybody comes from
So it doesn't.
No, it doesn't matter. People from Britain use different letters and different pronunciations. People from here use different pronunciations. It changes. Going into some sort of scientific convention, you better be able to adjust to slight differences on everything. Okay? All right. Now, coming back to the next spore stain. What was the other spore stain? stain you use to stain the spore in that case? Not saffron. Carbofuchsia. But yes, it's still red. Yes, it's still red, but it's not saffron. So carbofuchsia stains the spore, drive it in. You then switch to a negative stain. What was the negative stain? Nigrosin black. So the background should be dark. The cells should be white to light gray, maybe a little bit of overleakage or a little bit of a light pink. The spores should be dark red to black red. Bright red to dark red to black.